So yeah, I'm Larissa. I'm a PhD student in Zurich, the University of Zurich, and I work under the supervision of Christian Grimm. And I'm working on my thesis mostly on hypoxia. And today I would like to discuss with you the metabolic consequences that we discovered in photoreceptors when they are exposed to chronic hypoxia. Just use a pointer. So to give you an overview of what I'm going to discuss today, I just wanted to already give away what the results are going to be so that in case anyone gets lost, they didn't miss too much. So what we discovered that in wild type rods, so just healthy rods that are exposed to chronic hypoxia, that before they degenerate, they actually have changes in the redox status, which we found because we saw oxidation of nucleotides, a protection, increased protection against ROS, and increased glutathione transferase activity. And we saw changes on fatty acid biosynthesis. But to be able to get to all these conclusions, I will discuss first the metabolic landscape of the retina, so everyone is on the same page. Then I would like to discuss the disease model that I've been working with, and then eventually I want to discuss the outcome and the results that I just described. So when we look at the retina and we talk about the metabolism of the retina, we now have this hypothesis that we have a interaction of the photoreceptors with the RPE, and that somehow the different metabolic processes work together between these cell types to get a healthy environment. And when we look at the mitochondria, we know that in the photoreceptors, they are mostly located in the inner segment of the photoreceptors. And this we were able to confirm in our reporter mouse models, where we could see very nicely in the cone mitochondria, the cone mitochondria are in the inner segment, and that they very nicely follow these elongated mitochondria follow the cell body of the cone. And then we have these punctated mitochondria at the bottom in the synapses. And the same could be said for the rod mitochondria, where we had elongated mitochondria in the outer segments, then we get punctated mitochondria in the ONL and at the segments. So then to study exactly what is the function of these mitochondria in the photoreceptors, we studied the interaction also of the RPE and the photoreceptors. So the basic hypothesis that we have now is that glucose is taken up by the RPE, and even though the RPE could use this for glycolysis, right now it's been said that the glucose is actually transported to the photoreceptors, and the photoreceptors use it for energy production. And the lactate that is then produced by the photoreceptors can be taken back up by the RPE, and this is then turned into pyruvate and used in the mitochondria for the different metabolic processes that are required for the RPE homeostasis. On the other hand, we have the daily outer segment renewal of the photoreceptors, and this gives us fatty acids, and these fatty acids can be used for beta oxidation and again then used for RPE homeostasis. So how does this then look more on a biochemical level? So when we look at basic energy metabolism, glucose enters glycolysis and follows this enzymatic pathway to produce pyruvate. And this whole process gives us 2-ATP. Pyruvate can then either be turned into lactate or into acetyl-CoA, and then it enters the TCA cycle and it can perform oxidative phosphorylation inside the mitochondria, which gives us 34 ATP. What's interesting about the photoreceptors, of course, is that actually they prefer glycolysis, and they have this process called aerobic glycolysis, while they do mostly glycolysis, even though they have the presence of oxygen instead of oxidative phosphorylation. While this is somewhat counterintuitive, because we know that the photoreceptors have a high energy demand, so they have a high need for ATP, we also know that they have a high demand for outer segment renewal. And what we know from glycolysis is that they have these branches, side branches, coming from the different metabolites in the glycolysis pathway that lead into the pentose phosphate pathway, into lipid biosynthesis and amino biosynthesis, which can then help sustain the photoreceptor survival. However, it doesn't mean that there's only glycolysis being done in photoreceptors. They still also use the TCA cycle, because also from this TCA cycle, we can make a multitude of amino acids and lipid biosynthesis. So this is what happens in the healthy conditions. And within my project, I am focusing on age-related macular degeneration. And for this, I'm mostly focusing on hypoxia. So what we know in a normal situation, so in healthy humans, we have a perfectly well-developed choroid, and the oxygen will diffuse to the photoreceptors. So what does that mean on cellular level is that we have HIF1 alpha, which is hydroxylated, and this can then form a complex with von Hippel-Lindau protein, and this can then be ubiquitinated and degraded by the proteasome. So this means that we will have no HIF1 alpha activity inside our healthy photoreceptors. So what we hypothesize is happening in AMD patients, or patients that have a low oxygen environment inside the photoreceptors, is that because we have a not so well developed choroid anymore, and we have this thickening of the Bruch's membrane, we have less oxygen diffusion, which results in a low oxygen tension around the photoreceptors. What this means is that HIF1 alpha cannot be hydroxylated, will form a complex with HIF1 beta, and will be transported to the nucleus, where it can be used as a transcription factor for multiple processes, promoting, for example, glycolysis and other important targets. So how I'm studying this within my project is we are using a VHL knockout that is rod specific. So what that means is that we have a well-developed choroid in these mice. We even have normal oxygen diffusion. However, we still have photoreceptor generation. And this is because in our model, even though we get hydroxylated HIF1 alpha, since we have a knockout of the von Hippel-Lindau protein, it cannot form this complex that can be degraded. So HIF1 alpha will still form a complex with HIF1 beta and can then be used as a transcription factor. So how does this look like in the mice? So this is work that's been done by the PhD student before me, and she characterized the degeneration of this model. And we could see at 10 weeks that we have still a very nicely developed ONL compared to control. However, if we look at 6 months, we see that we have degeneration of the photoreceptors because we have thinning of the ONL. What is important to note about this model is that we do not have a perfect expression of the Cre, so it is a bit patchy. So this can mean that we underestimate our results and our outcomes in certain cases. She then also studied the metabolism of these cells, and she did this by using a rod-specific lactate sensor, which she injected into these mice, and then she measured this sensor ex vivo with a 2-photon. And when she blocked the lactate transporter, she found that you get an accumulation of lactate over time in the VHL mice that is higher than the controls, suggesting that there is an increased production of lactate inside the VHL rod photoreceptors. This can then either mean we have an increased glycolysis, or it could mean a metabolic shift. So for this, she checked the glycolytic rate with another sensor, and she found there's actually no change in glycolytic rate in our controls versus the VHL mice. And this then clearly suggested that there's not necessarily more glycolysis going on in these mice, but that instead of the glucose feeding into pyruvate, it is now feeding all into lactate. So we really get this metabolic shift. To then further study the metabolic changes of these mice, she performed proteomics at 10 weeks, so that is before degeneration, and we were able to separate the photoreceptor into photoreceptor segment versus ONL. And what we could see is that in the proteome of the photoreceptor segment in the VHL showed an increase of preparative glycolysis enzymes. This makes sense because they all have an HIF1 alpha target site, and this then suggests that we have an increased glycolysis activity inside the photoreceptors. She then also saw that we have an overall decrease of TCA cycle enzymes in both the segments and the ONL. And lastly, she saw a complete downregulation of oxidative phosphorylation proteins inside the photoreceptor segment, so that there's a very severe downregulation of OXPHOS activity inside the Rod VHL mice. However, for the ONL segments, she saw an increase of these proteins, which could suggest that maybe the mitochondria are also traveling towards the ONL instead of staying in the segments. So based on this data, we hypothesized that maybe the lack of oxidative phosphorylation inside our Rod VHL mice is what is causing the degeneration. So because of this, we developed a mouse model that has a COX-10 knockout inside the rods, which would lead to a complex 4 dysfunction, since COX-10 is an assembly factor of complex 4. And as you can see, COX-4 is downregulated in the rod COX-10 mice, even though it's patched because of the CRE. However, both at 3 months and 6 months, we didn't really see a proper degeneration of these mice, and they didn't show any lack of activity or ERG or anything else. So from this, we concluded that it's not the lack of OXPHOS and the lack of ATP production that is causing degeneration in wild-type photoreceptors that are exposed to chronic hypoxia. So then, this is where my PhD started. So then is the question, what are the metabolic changes that actually precede photoreceptor generation, and why does the response to hypoxia kill? So to start this research, I went back to the proteomics data that we have. So this is, again, our mice, our retinas were extracted at 10 weeks, and we separated them for photoreceptor segments and outer nuclear layer. And this time on the proteomics data, I excluded the proteins involved in oxidative phosphorylation and glycolysis, just to make sure that they weren't masking any other results. And based on this, we got a multitude of proteins that were up and down regulated. And since it was mostly hypothesis testing and trying to figure out which possible pathways could be involved, for now, the gene counts aren't too convincing, but we just disregard them for now, since we were just trying to see in which direction we should work. So what we saw were proteins that were upregulated in photoreceptor segments was mostly glycogen biosynthesis. We saw a downregulation in purine metabolism in the photoreceptor segments. Furthermore, in the outer nuclear layer, we saw an increase of the nitric oxide transport and hydrogen processes, and an increase in glycogen. And in the outer nuclear layer, we didn't really see many proteins that were downregulated. So what does that mean? So when we then look at the photoreceptor segments, we saw this strong increase of glycogen biosynthesis, which could be explained that the cell is starting to get into a stress state and trying to store energy for the time that it runs out. And we saw a change in purine metabolism. When we then studied the pentose phosphate pathway, we have this gene G6-PDX, which is the entry of the pentose phosphate pathway in mice. And we saw a decrease of this on RNA level at 10 weeks already. So this already suggested that maybe something is happening in the side of the purine, or maybe even the redox status of the cell. When we then looked at the outer nuclear layer, we saw very clearly proteins that were upregulated that are important for the protection against reactive oxygen species, and all glutathione S transferases that we were able to capture with the proteomics were increased, suggesting again that there is a disbalance in the redox status. When we then looked at activity of specific enzymes, we saw a decrease of aconitase in our VHL mice. Aconitase is an enzyme of the GCA cycle. So of course we were already expecting this, because we have a metabolic shift towards more lactate production instead of pyruvate production. But also specifically, aconitase is also regulated by reactive oxygen species. So that could also explain the decrease of this activity. And then we also saw a trend of an increase of glutathione S transferase activity in our VHL mice. So this was all done on whole retina. So the fact that this is not necessarily statistically significant could also be that there is so many other cell types from the inner retina that are also affecting this data. So we could still see a trend suggesting that the effect is quite strong. So to then add on to this data, we wanted to perform metabolomics to see if on the metabolic level and metabolic composition, we could see any changes between our rod VHL and control mice. So again, at the exact same time point, we separated the photoreceptors and the outer nuclear layer, and we performed metabolomics. And here we can see on this metabolic heat map that the VHL mice, so this is the photoreceptor segments and ONL areas combined, that we could see a nice separation of the two. Another thing that is interesting to note about this heat map is that you really see within the VHL flocks and the VHL opsoncrine two groups, and this is a sex difference. So there also seems to be a sex difference in these mice. However, we would need bigger numbers to be able to really identify what the sex difference would be inside this genotype, which we were not able to do so far. And when we then compared the metabolites and we put them in the volcano plot, we could see that there were a multitude of metabolites that were up and down regulated in our rod VHL versus the controls. So if we then look at the most abundant metabolites that were affected, basically what was very, very obvious is that we have a strong decrease in the nucleotide pool inside our VHL mice. So this could be either due to decreased production or increased degradation. And I've discussed this also with Dan, and he has done experiments where we try to trace the pentose phosphate pathway inside the retina, and we could actually not really see a strong activity of the pentose phosphate pathway inside the retina. And this would suggest that it is more about increased degradation of the nucleotides inside the VHL mice. What I think is happening is that because we have this strong redox balance, there is oxidation of the nucleotides, and that of course changes the metabolic structure of the chemical structure of these nucleotides, which not allows us to measure them as these nucleotides, and that's why we see a decrease. So the current hypothesis that we are going for with this part of the story is, since we have this increase of reactive oxygen species, we get this redox imbalance inside the cell, which is then affecting the nucleotides and is increasing the glutathione transferases. But this is, of course, not necessarily an immediate cause for degeneration. So then, of course, the next question is, okay, we have the presence of ROS, we have a redox imbalance, but why are these photoreceptors degenerating? So for this, I worked together with Dan again to study the flux of this retinas in the VHL mice versus controls. And basically what we did is, again, at 10 weeks, I isolated the retinas and I incubated them in a buffer that is supplied with 13C glucose. So for people who are not familiar with tracing experiments, glucose is a metabolite that has 6 carbons, and normally a carbon has a mass of 12. So since we now have a glucose that has carbons with a mass of 13, this allows us to measure metabolites, and when they have a mass that has a certain amount of mass is higher based on the carbons that they have, then we know that it's coming from the glucose that we fed the retinas, so that it's a synthesized metabolite, and this allows us to then trace which metabolites are generated at one point in our retinas. So we sampled then our medium at different time points, and this allowed us to have a steady state concentration. And we also we collected the retinas at 90 minutes. So what it means to have a steady state concentration of this 13C glucose in our sample means that what we will be looking at is exactly the total abundance of a metabolite, whether that's changed in condition A versus condition B. And what we could find is, when we look at the medium, is that there's actually no more glucose utilization or less in the VHL mice versus the controls. And there's also no difference in lactate production, suggesting that the glycolytic rate actually between the VHL mice and controls is similar. We then checked for the total abundance of the metabolites that were labeled by 13C in the whole retina, and we also couldn't really see any differences. While some metabolites do give a trend, there is no real differences, which could either be masked by the inner retina, or it just means that in the steady state, there is no difference between disease and control. To then continue our studies, we decided to do a dynamic flux experiment. So this works a little bit different from the steady state, since we now don't get a steady state glucose concentration inside, since we only expose the retinas to 13C glucose for 0, 1, and 5 minutes. So what does this mean is that we're not looking at the total abundance of the metabolites, but we're actually looking at how fast are metabolites labeled. So that means if we have a faster labeling of a certain metabolite, it means that the enzyme is probably working a little bit faster. And when we look at this data, we actually saw an increase of the labeling of in the rod VHL mice in DHAB, 3PG, and PAP, and on the other hand, in glycerol, 3P, and glycerol. So what this then kind of suggested, or what we hypothesized, is there must be some changes on the fatty acid biosynthesis inside the VHL mice. So then I tried to explore more the fatty acid biosynthesis, and I checked the pathway that is involved in the synthesis of fatty acids, and we saw at 10 weeks, so again before degeneration, a strong increase of on RNA level of different enzymes that were involved in this pathway, such as SCD2 and CPT1A. Then we also have the metabolomics data, but since our metabolomics data was based on polar metabolites, we only captured this lauryl carnitine, which seemed to be increased. But so for the future, it would definitely be good to look at the lipidomics and have a chance to look at non-polar metabolites to get more information on how fatty acids are affected. So then to conclude the second part of the story is what we saw is in our VHL mice, we basically have an increase of enzymatic activity that allows a higher flux inside the lipid biosynthesis. So we then also, because of course we are really focusing on photoreceptors inside this whole story of AMD. However, it's also important to look at the ICUP, or at least the RPE. So what we saw here is when we did, again, the dynamic flux inside the RPE, we saw that even though not significant, we saw a trend of an increased metabolite labeling in the VHL mice compared to controls. So my hypothesis here is that the RPE is trying to increase metabolism to somehow support the photoreceptors, since they seem to be slowly starting to dysregulate. So then to combine all of this information, what we can conclude so far from my studies is that in wild-type rods that are exposed to chronic hypoxia, we see a change in the redox status and in the fatty acid biosynthesis, and this is all happening before degeneration, suggesting that this could potentially cause degeneration, but this is something that we still need to properly study. So what does that look like on a more biochemical level is that we have about the increased presence of HIF1 alpha. This causes an increase of reactive oxygen species. This will somehow affect the redox status and also the lipids, and this then causes an increased flux into the lipid biosynthesis to somehow support the outer segment renewal. So, of course, we are not done yet here. So the idea is to now properly determine the levels of nucleotide oxidation and to really complete the story on the redox status, to perform lipidomics to further study the lipid composition of the photoreceptors. And it's also a question, if we would scavenge the reactive oxygen species, would this then rescue the phenotype? So another thing that I think is very important about this story is that every single treatment or therapy or this kind of things has two sides to a story. So I've shown here in my thesis or my PhD that when we knock out VHL in healthy rods, this causes degeneration in the long term. However, there was a paper published this year by Professor Zhang and his group that if you knock out VHL in retinitis pigmentosa model, you actually see survival of the photoreceptors and less fast degeneration. And when you look at their tracing data, because they also performed the tracing, you can see very nicely that they see an increase in glycolysis. So what I'm taking away from this is that while we get this increase of glycolysis, which then allows increased production of these metabolic intermediates because of the fatty acid synthesis and amino acid synthesis, while this could protect short term, because we clearly see it in this RP model, potentially long term, it could cause problems because then you completely dysregulate the metabolism of the cell. So I think it's a very interesting when we want to modulate metabolism that it's unfortunately not as simple as we modulate one pathway and then it's done, that we need to be mindful that long term there could be consequences when we start to play with metabolism. And with this, I would like to conclude my talk. I want to thank Christian for always supervising me and everyone else in the lab for supporting me. And I want to thank Hurley Lab in Seattle for allowing me to go there to perform these tracing experiments and to teach me everything about metabolic flux. And just to, I don't know, promote myself a little bit, I'm also graduating in 2026 and I would like to do a postdoc in metabolism. So if anyone has any options or wants to talk, I'm very happy to have a conversation. So that's it. Thank you.
Hi, so awesome talk. I really think you put this data together in a way that I think made more sense than any time I've heard it before. So I don't know, I mean, it's coming together really well.
Yeah.
I think I'll just start off the questions and other people either drop questions in the chat or if you raise your hand, or just talk, that should be fine. Maybe raise your hand because somebody might be raising their hand and then somebody might be talking. Anyway, so I have a few questions, but I'll ask one for now. Is there any effort to actually measure reactive oxygen species? I mean, this seems to be kind of a central component of your hypothesis. And there are a lot of people who mismeasure reactive oxygen species, but I mean, there are definitely ways that you can do it and even look at the dynamic production of something like H2O2 over time.
Yeah, so this is something I would like to do, but the protocols that I've seen so far, I didn't really believe that it would be very useful. So it's still something that I'm trying to research and trying to find the right protocols for, because there are a lot of them out there, but some of them are less convincing than others.
Totally, totally. I mean, yeah, I've been down that path myself. I can message you about what I think of protocols I like. I mean, I don't know if you could also not buy those.
Yeah, because I
Uh,
know there's the inside the Ouroboros system, you could potentially measure it, but this I'm not sure because
the
it will
ret
also completely destroy everything.
yeah, the retinas get so torn up. I mean,
Yeah, because I have tried this system
you know,
and it didn't really work very nicely.
yeah, I'll email you. Francois.
Yeah, thank you for this nice talk. I hope you can hear me. So this one set of data where you were showing that the nucleotide pool was going down, at least the number of metabolites indicating that. And you said that this might be due to oxidation of nucleotides. Could it also be due to the consumption of these nucleotides for energy generation? The nucleotides,
Yes,
after all, are relatively energy rich.
Yes, this is still possible. So I'm now still optimizing the protocol to measure the nucleotide oxidation. So not, right now, that's my working hypothesis. So I'm doing the experiments, and hopefully in a few weeks, I will have the answer. And yeah, it's still a good suggestion. It could also just be utilization.
And then another question. After this dynamic flux with carbon tracing, you found that there was no change in glucose consumption when you measured the glucose in the medium. Did you also measure the lactate release? Was that perhaps
Yes.
changed?
No, so that was also unchanged.
Okay. Okay. Well, then I have no question to that, though. Thank you.
Thank you.
Claudia.
Hello, Larissa. Very nice talk. Have you thought, which is kind of a longer term experiment, to cross the HIF1 alpha conditional with your VHL conditional to actually really determine if it's caused by HIF1 alpha or not something else, right?
So we have this mouse line, so that's something we could potentially do.
Especially, in regards also to the other paper from Stephen Sung that you mentioned at the end, right? VHL also regulates AKT activity and increases AKT activity, decreases AKT activity. So it could be that you end up with changes in AKT and regulating metabolism in a completely different way, which could also explain, other results.
Yeah, so we have this mouse line, and we have done different experiments, not necessarily focusing on metabolism. So it's definitely something we can try and see if we get the similar results or completely different. Thank you.
I'll take the opportunity to ask more questions. I have a couple pretty simple ones. One, do you have a sense of whether or not there's any variation in the level of patchiness of the Cree? Really beautiful Cree image, by the way. I think that's the first time I saw that one.
Yeah, we have a new microscope, and it's really nice. Um, so we're not really sure. So we think it's about 50% that we have Cree expression, but the variation between the mice we don't know.
Okay. I mean, that might be really important for being able to see a signal in a lot of these experiments that we're doing where, we're just homogenizing a whole tissue to get these flux measurements, especially. So knowing if in some samples there's effectively zero knockout or little knockout and some there's a lot, it might be good to try to filter samples that way somehow.
Yeah, so we've tried to determine the variability based on qPCRs and trying to do different experiments, but so far it's not really been successful to determine really the level of Cree.
Okay. And I'll keep asking until someone raises their hand. Glycogen. I mean, you saw this kind of hint that glycogen would be, strongly changed. And then it didn't, I mean, it's not going to pop up, I think, in conventional metabolomics experiment, but is there any intention to measure on its own just levels of glycogen in the retina? And I mean, that was quite interesting.
Yeah, so far I've not really been focusing on the glycogen part, but that's definitely something we can look into and see what kind of experiments we could do there to kind of complete the story.
Okay, then I have one more question, and this is maybe difficult to answer, something that you probably don't have data on, but maybe you know the literature about this. So the retina itself, I mean, Rob is here and he can comment on how low the oxygen tension is, at least in some parts of the retina, like the inner segment, and oxygen tension should be the driver for hydroxylation of HIF. So you'd expect that there's fewer molecules in the sort of relatively hypoxic inner segments. There are fewer molecules of oxygen to hydroxylate HIF. And so I guess the question is, why is HIF not chronically active in a photoreceptor? could it be because there are different kinetics for hydroxylation in the retina versus in other tissues?
That is a great question that I don't have an answer to right now.
Yeah. I mean, I guess the question I really wanted to ask is about whether or not the hydroxylases, whether there's some variation, some modifications that can be made to those that would alter their sensitivity to oxygen.
I can ask another question. And that goes to this COX-10 knockout, rod-specific knockout, I think. So can you? Yeah, I'm not too familiar with this particular isoform of cytochrome oxidase. Can you elaborate a little bit more on what is missing then, and how this would affect metabolism? And could this subunit of cytochrome oxidase somehow be replaced by another one?
So this COX-10 is a protein that allows the assembly of complex 4. So it would basically mean that complex 4 cannot be established. So you have a knockout of complex 4, which would then just completely, at least more or less completely knock out oxygen phosphorylation. So it could, of course, in oxygen phosphorylation, it is not that straightforward that you go from one complex to the other, and it could skip certain complexes. But we also have injected other mice we have studied other mice where we've knocked out COX-10 and other cell types. And here we did see a change on the phenotype. So this kind of gives us the idea that it is functional, and that the COX-10 knockout does work, but just for the rods it doesn't. That we don't need to.
It doesn't cause degeneration.
Yeah.
Okay.
Well, maybe I can add to this a little bit. So we knocked out COX-10 from rods, cones, RPE, and inner retinal neurons with different phenotypes there. So each of these cells seem to respond differently to the lack of COX-10, whether this is really due to the lack of OXPHOS or whether this is due to differential usage of or need of COX-10 for building up complex 4 in the end in these cell types, we don't know yet, but there's clear differences between cell types. And for the rods, why they don't die is because they clearly do lots of glycolysis or aerobic glycolysis. We just have this paper now submitted. So they can produce energy, and they probably still have some functional TCA cycle left because the oxidative phosphorylation comes later, although the two talk to each other, but some TCA cycles left, so they can build up sufficient building blocks for the renewal of the outer segments. That's probably keeping them alive because they can really go back to ATP production via aerobic glycolysis and still have sufficient supply otherwise, whereas other neurons in the retina really depend on OXPHOS, and knocking COX-10 out of those, then these cells are in real trouble. That's the current hypothesis about what we have.
May I ask then, when you said you did a conditional knockout in rods and in cones of COX-10, were there any particular differences between rods and cones in the response to this? We may have talked about this before, but I've forgotten.
Well, it's hard to tell yet. We haven't had the chance to analyze many mice at this point. We just know that, for example, if you knock out COX-10 from cones, they die not quickly, but in 1-year-old mice, you have lost, I don't know, 70% cones. Well, the cones at least that express Cre, so to say. But we don't have kinetics on that. We don't know exactly whether there is a strong difference between rods and cones. I do believe so, but I cannot, it's no hard evidence yet.
Okay, thank you.
Abby.
Thanks. Hi, Larissa. It was a great talk. I was wondering if you've ever tracked lipofuscin production and if you see a greater production in the VHL mice compared to the age-matched controls?
No, this is not something we have done yet since we only just restarted the whole fatty acid hypothesis, but that's something I could definitely have a look into to do. Thank you.
And kind of another comment on this. I like that the orientation of what metabolism, of the theory that metabolism is doing a lot of sort of biosynthesis in rods, both mitochondrial and sort of glycolytic metabolism are oriented towards that. Do you see, though, with these changes, any difference in outer segment? I guess it would be really tough to measure turnover, but just maybe length. Do you see them getting sort of, at least before degeneration, could they be longer in these VHL mice?
So this is something we haven't really properly looked at yet. So that's something we can definitely do. I did try to see if maybe the mitochondria itself before degeneration have different morphology, but based on just a confocal microscopy, we couldn't really yet distinguish between the rods, VHL, and the controls.
You're up.
Hello. I have a question about this model. So I think your model's differences with the real hypoxia is the oxygen level is still high, relatively high, right? So would that be the cell death because the HIF-alpha was up and then was stabilized, so the anti-oxidation reaction is low, but there's still high oxygen, so it's
Yeah,
more
so that's
toxic to the cell. So have you seen any antioxidant pathways changes?
Yeah, so that's something we've also been thinking about because, of course, we don't have the oxygen utilization since we completely now skip oxidase phosphorylation, and that this high presence of oxygen could then also affect the cells. So within the model, this is definitely possible. I think if we think in like the grand scheme of things, if we still think about the model, it could still be useful because we know that metabolism is affected and we will have this decrease or changes at least in the glycolysis inside our disease, like in the context of AMD. But yeah, within this model, it could definitely be that the high abundance of oxygen that is not being used could cause some problems inside. Yeah.
Yeah. Another question is in one of your data that the statist glucose labeled glucose tracing experiment, why the lactate is not higher?
Good question. Yeah, I was also surprised because I was also expecting that if we have an increase in glycolysis, that would mean, because we saw this metabolic shift in previous data, that it would mean an increased flux into the lactate. So my guess is now that the glucose is feeding into the fatty acid biosynthesis and that we can see it there, and then maybe it doesn't reach as fast for lactate, but this is still something that I'm not sure about, and that is, yeah, I can't really explain for now. Since we know that glucose splits into these 2,3 carbon molecules, it would make sense if then also the other side of the pathway would be increased. But for right now, yeah, we don't see this. Could also be potentially because it's masked. Maybe if we look at the other metabolites, they have a higher production in the photoreceptors compared to inner retina. That's why we can see the differences better, and that maybe lactate production in inner retina masks the data or the measurements or the excretions from the photoreceptors, but this I cannot
Thank you.
confidently say.
I just want to also mention that the pools of some of these intermediates that get measured, DHAP, glyceraldehyde 3-phosphate, or glycerol, or they might not be nearly as high as the pool size for lactate. So it might be easier to push that pool around a bit and make it look like there's a big change. But lactate is just so abundant that even if you have sort of a few more molecules flowing through it and you're seeing a change in labeling of glyceraldehyde 3-phosphate, for example, you might not see it in lactate because there are just so many molecules of lactate to start off with. Just sort of noting that possibility, I guess. If there are no more questions, then thank you all for coming. And thanks, Larissa, for giving a wonderful talk.